Hello and welcome to the With My Plant channel. Before starting the class, I want to wish you a very happy new year. आपका नव वर्ष एक स्वस्थ एवं एक खुशहाल भरा वर्ष रहे. So today we are going to start our next series of lectures. It is on gibberellin. It is on gibberellin. And the first lecture on this series is the discovery of gibberellins. Okay, so let's start. So if we see the history of gibberellin, the gibberellin was discovered from one disease called foolish seedling disease. The foolish seedling disease, otherwise it is known as bakanae disease. In Japanese, as it was discovered in Japan, so the disease was called as bakanae disease, and the symptom was basically it was growing tall, but it was not producing any seed. The rice seedlings, the rice farmers actually saw this problem, and they found that it is growing tall but did not produce any seed. They called it as a foolish seedling disease. Okay, so scientists studying this, they found that a fungus infected in these tall plants and made them grow extensively, and they isolated the fungus and the chemical secreted by the fungus, which is responsible for this tallness of the rice plants, and name it as gibberellins after the fungus Gibberella fujikuroi. This is the name of the fungus Gibberella fujikuroi, on which the name of the chemical was named. Okay, so in 1930s, Japanese scientists got some crude crystals of two active compounds called as Gibberellin-A and B. Okay, but because of communication issues, the World War, because of this World War second, there was lots of communication issue. That's why they could not convey this till 1950s. So after 1950s, it came to Britain and USA, and they came to know about this chemical. Finally, scientists in Britain and US succeeded in understanding the structure of gibberellic acid in the middle of 1950s. At the same time, researchers in Tokyo isolated three types of gibberellins from gibberellin A. Okay, and they realized that there is a whole family of gibberellin, and each fungus culture produces different ones. Okay, with gibberellic acid being a primary one, all gibberellins share a common feature in their structure derived from end cowrin ring. So this about this end cowrin ring we will understand in the biosynthesis class. Biosynthesis, this is very important in biosynthesis. We will understand this ring, about this ring, in biosynthesis class. Okay, when scientists tried gibberellic acid in various plants, they saw remarkable effect, especially in making the plants dwarf. Okay, or dwarf plants grow taller. Okay, so both way it was acting, or basically this is working in tall, making the plant tall, but the genes involved in this can be modified to produce this dwarf plant. Okay, however, genetically tall plants did not show much change with gibberellin. This showed that natural plant growth is regulated by gibberellin. Okay. After discovering the growth effects, gibberellic acid, scientists found that similar substance in different substance in different plants. Actually, what is happening? Uh, there is a basic structure of gibberellin, and in different plants there are some amount of changes, small changes in on that basic structure. That's why the substance, they will act as a gibberellin or act equally, but the structure are a bit different. Okay, that's why there is similar substance, not same, but it is similar substances. These substances act as gibberellin but did not have their exact chemical structure. This hinted that there were gibberellin like compounds even though their structures was not fully defined yet. So in the previous slide, we found that there is no any scientist. Okay, so but exam point of view, name of the scientist is very important. That's why I collected some more information, which is placed in second slide, where the important scientists or the contribution of important scientists is listed. So in this slide also, here in this picture, we can see the symptom of bakanae disease. This is a healthy seedling. This is an elongated seedling or infected seedling basically, and this is a stunted seedling. So you can see there is a huge difference in the seedling's length. Okay, and this is because of this fungus, Gibberella fujikuroi. Okay, so coming to the list of contributions of scientists, one scientist was there, Hori, in 1998 suggested fusarium as the culprit of this bakanae disease. Then Savada, another scientist in 1912 hinted that this fungus might secrete something responsible for the illness. The discovery by Kurosawa was very important. This scientist's name is very important. Kurosawa in 1926 attributed the disease to substance emitted by the fungal species, this Gibberella fujikuroi. Okay, so this is very important. Sparkling debates over the actual pathogen. Then Olin Weber in 1931 linked the fungus Fusarium moniliforme, another fungus, to the Ascomycetes Gibberella fujikuroi. And Yabuta and Sumiki in 1938 breakthrough involved isolating a pure crystalline substance, naming it as a Gibberellin-A. So Yabuta and Sumiki, these scientists, they basically um, isolated Ga in crystallized form. Okay, so this is important, that who has isolated the chemical or gibberellin, and another question may come, who was the one who detected that the disease is because of this fungus Gibberella fujikuroi. Okay, then further discoveries followed. Takahashi isolated GF4, which is a bioactive GA, from the fungus in 1957, while, Macmillan and Suter identified GA1, another bioactive gibberellin, in 1958. So GA4 was first isolated and GA1 was secondly isolated. Cross and colleagues in 1961 isolated 6 gibberellins from different fungus, that is Fusarium moniliforme, naming GA, GA1, GA2, GA3, GA4, GA7, GA9. To avoid confusion, Macmillan and Takahashi 1968 pronounced numbering of gibberellin symmetrically. Additionally, Macmillan and team in 1961 isolated J5, 6, 8 from bean seed. This is important. From which plant which GA is isolated is very important. Then Mulholland's discovery in 1963 added GA 10 and 13 to the list. So over 125 it is written, but around 300 or more than 300 gibberellins have been identified till date. Okay, so these are the important scientists such as Macmillan, Takahashi, then this Yabuta and Sumiki, Kurosawa. These are important scientists you have to remember. And GA1 and GA4 are the bioactive GAs. And this number which is attached with GA is called as A number. Okay, we will understand it in next other classes also. A number, it is not exactly, but basically based on the sequence of the discovery. Okay, so if we see the structure of gibberellin, it is having a basic structure. On that, modifications are there, like that that basic structure is called as and gibberellin, and it is having one, two, three, four rings. Okay, we will understand here. In 1958, scientists identified a specific gibberellin, that is gibberellin 1 or A1, from runner bean seeds. Okay, this is very important. They found that immature seeds had much higher concentration of gibberellin compared to other part of the plant. However, because gibberellins are usually present in every low amount in plants, in very low amount in plants, in around 1 to 10 parts per billion, that is ppb, in vegetative tissues, and up to 1 parts per million in seeds. Okay, seeds में थोड़े ज़्यादा होते हैं as compared to your shoot. Okay, chemists needed large quantities of seed for extraction. As more gibberellins were discovered from both fungi and plants, they were numbered as gibberellin A 1, 2, 3, like that. That is J1, J2, J3, like that. X. Okay, unknown. This X is unknown. In the order they were found or they were discovered. This numbering system, established in 1968, helped organize and avoid confusion in the naming of the Gibberellins. However, it's important to note that numbering system does not imply closely chemical similarity or metabolic relationship between Gibberellins and adjacent numbers. All Gibberellins share a common backbone structure based on the end Gibberellin structure, which is this end Gibberellin structure. This structural similarity defines them as members of Gibberellin family despite their diverse numbering. Okay. Essentially, within the family, Gibberellins, some contain the full set of 20 carbon. This is very important. Some contain 20 carbons, while other contains only 19 carbons due to metabolic process where one carbon is lost. Most of the bioactive gibberellins, if we see, these are your basically 19 carbon compounds, such as your GA1 and GA4. Okay, there are variation in the basic structure, particularly in oxidation state in carbon 20, and the number placement of hydroxyl groups of the molecule. Despite the number type of gibberellins found in plants, genetic studies have shown that only few of them actually act as active hormones. The rest either serve as a precursor to active forms or exist in inactive version. It means the 20 carbon GA are basically precursor of these bioactive GA, and these, in the process of forming bioactive GA, they used to lose one carbon. Okay, so this is all about your discovery of gibberellic acid. I hope you got the idea of the discovery and the basic structure of GA. I have tried to compile it in gist so that you can easily understand and remember. So thank you for patient listening. and please share, like and subscribe my channel, and share the video as much as possible who need it actually. Okay, thank you.